Hi everyone, we're going to talk about the complement fixation and neutralization reaction. So we're going to start with complement fixation. So it was developed by Jules Bordet and Octave Gengo, and it measures antigen antibody reactions. It is a test for infection with a microorganism that involved measuring the amount of complement available in a serum to bind with an antibody antigen complex.
So in complement fixation, the body's natural defenses use complement to bind to antigen antibody complexes, helping to destroy those pathogens. Molecules and component involved in this include sheep red blood cells, antigens, antibodies if present, the complement, and the Ag/AB complex. The complement activated by combination of antibody complexes. So the protocol to do complement fixation starts with heating the serum at 56 degrees Celsius to destroy the patient's complement. A measured amount of complement and antigen are then added to the serum. If there is presence of antibodies in the serum, the complement is then fixed due to the formation of AG-AB complex. If no antibody is present, then the complement remains free. To determine whether the complement has been fixed, sheep red blood cells and antibodies against sheep red blood cells are added. In the positive test, the available complement is fixed by AG-AB complex and no hemolysis of sheep red blood cells occurs. The test will be positive because of the presence of those antibodies. In the negative test, no AG-AB complex reaction occurs and the complement is free. The free complement then binds to the complex of sheep red blood cells and its antibodies cause hemolysis, causing the pink color of the serum. So the principle is that the nature of the complement is to be activated when there's formation of antigen/antibody complex.
So it was created in 1901, and serums from individuals suspected of having typhoid fever were combined with typhoid antigens. So this demonstrated a positive fixation of complement in cases of typhoid fever.
So current uses for the test include to detect the presence of any small traces of specific antibodies or specific antigens in the patient's serum. Companies that use this test include laboratories or clinics such as LabCorp, Quest Diagnostics, and Alere. Such companies use the test to specifically diagnose patients for viral, fungal, and rickettsial diseases such as rubella, herpes, simplex virus, and other diseases.
So it can be used for COVID-19 testing. There was a study in the late 60s, early 70s on a different strain of coronavirus. And the study was on normal adult sera was taken and tested by complement fixation against 3 different coronavirus agents. So because coronavirus complement fixation has a relatively short half-life, this test can be conducted, but it's not the best test to use.
Now we're going to talk about neutralization reactions, and these basically detect bacterial toxins and antibodies to viruses. So for some background in history, during the late 19th century, Emil Behring and Ehrlich were 2 scientists that discovered neutralizing antibodies. Neutralization in general refers to the ability of antibodies to be able to block the site on bacteria or viruses, which the virus will use to enter the target cell. So by inhibiting or neutralizing the antigen, they're able to inhibit it from having any effect, which is why they're known as neutralizing antibodies. And diagnostically, the evaluation of these neutralizing antibodies can be measured through a neutralization reaction.
Now we're going to talk a little about the key principles of a neutralization reaction or test. So the parts are cells, the toxin or the virus, and the antibody or the antitoxin. And the antibody sera is put into a dish with the virus or toxin and cells to see if the antibodies can neutralize the virus. And as you can see in this picture, the toxin is kind of covered by the antibodies, and then you could tell if the test worked by seeing if the cell is damaged by the toxin or not in the end.
To go more into the protocol of how the test is actually done, first the patient's serum is mixed with the suspension of infectious virus particles of the same type as those suspected of causing the disease in the patient, and then a control suspension of virus is mixed with the normal serum and inoculated into cell culture. And if the patient's serum contains antibodies to the virus, then the antibody will bind to the virus, preventing them from invading the cell culture. So as you can see in this image, for row B, the patient doesn't have any antibodies, so the virus is able to bind to those red blood cells and cause agglutination, which would be a negative result, meaning the patient doesn't have the virus. And in row C, the patient has the neutralizing antibodies, so they bind to the virus, preventing any effect on those red blood cells. So this would mean that the patient is positive for this virus.
So the usage, it was first used by Emil Behring and Paul Ehrlich in the late 1800s, and what they were trying to identify was antibodies for the diphtheria toxin. And it's still used today, and it's mostly used for influenza, and it's also used to make vaccine anti-sera, which is how our flu shots are made. And one company that sells antibodies and assays, like the neutralization test, is Rockland Antibodies and Assays.
In regards to companies, its application is restricted to laboratories that perform routine viral cultures and related diagnoses. These companies include List Biological Laboratories, which is a company headquartered in California, and it produces bacterial toxins, and their products are involved in vaccine development, toxin research, and infectious disease research. And other companies include LabCorp and Nelson Labs. And in regards to COVID-19, although not currently used for COVID testing, a neutralization test would be able to indicate whether the patient has active antibodies against the virus.